This is a production of Cornell University.
Sure, thank you, Rochelle. So good afternoon, everyone. Today we have two research about breeding for disease resistance, but let's start with my non-edible switchgrass first. So my research is to improve switchgrass population to be resistant to diseases caused by Bipolaris or Ricey. And today I would like to introduce you what is switchgrass, because I don't think many of you know well about switchgrass. How many of you know switchgrass? Like literally touched switchgrass before? It's pretty itchy though, and I'm gonna tell you about background of the disease that I'm working on, the progress of the selections of those diseases, and the correlation between the resistance of two diseases. So this is me with switchgrass, and switchgrass is pretty newly domesticated crop. It was just domesticated just only less than 100 years ago. So we still have like lots of diversity in switchgrass. And it was only about like 1 decade ago that we use it for, that the Department of Energy has selected switchgrass as a biomass energy crop. So basically, switchgrass was just grown to like proposed to be the plant for producing electricity, and also can convert it to ethanol in the future. When the gas price is pretty high, not this time, but maybe in the near future. And you may think that, hey, in these pictures, like the switchgrass is even like smaller than me and like not that good in terms of the yield in these pictures. But in this picture, it was taken in around Penn State University, and this area is proposing to be an ideal area for planting switchgrass, because it's a marginal land, which means that no other crop can make any like good yield based on this in this land. Like in this area is a mine claimed area, which means that like nobody cares about that land, and switchgrass was pushed to plant it in that low marginal land. So, and most of the research that has been done in switchgrass, it's focusing on increasing the yield and other agronomic traits, but very few, just only couples of research has been proposed about the disease resistance in the switchgrass, even though one of the disease that can reduce the yield up to 50% of biomass, which is a whole world of switchgrass. If you reduce the biomass of switchgrass, like switchgrass is nothing for you. So these diseases that caused by Bipolaris or Ricey have a significant, not only for the yield reduction, but also it can reduce the germination, which switchgrass is a perennial crop. So the first year establishment is very important for switchgrass. If it can reduce the germination, which it means that it reduces the biomass that you can get for many years also. So in my breeding program, I'm working with upland switchgrass, shelter, and cabin rock. And based on that two diseases, Bipolaris or Bipolaris leaf spot and Bipolaris seed rot, I separate them into like two distinct breeding program. And I already done the 2 cycles of selection based on those traits, and I'm gonna continue the 3rd cycle of selection based on Bipolaris seed rots, and you will see like why I'm keep continuing this. And based on this 2nd cycle of selection, I test the reciprocal test to see the correlation between the traits. So let's move to the first resistance, the resistance to Bipolaris leaf spot. Basically, I just spray the inoculum on the 4-week-old seedlings and cover it with a clear plastic bag for 24 hours, release it and leave it for 7 days, and evaluate the disease based on the percent lesions on the leaves. And because this disease has very few studied previously, so I didn't know like the genetic background that control this trait before. So before you jump in general, before you jump in for breeding anything, you should have a background of this, how is this disease heritable. So I wanted to calculate the heritability before I made any selection. So I produced the half-tip seeds of the lowland type of callow in greenhouse, because I cannot produce the seeds in the field in Ithaca, too cold. And I produced the half-tip seeds of the callow and shelter in the field, and surrounded by surrounding them by the sorghum Sudan to produce to prevent the cross-pollination between the plots. But sadly, the heritability based on this resistance to bipolaric leaf spot is significantly non-significant or zero, which means that I may not expect any gain from selection on this, but I still hope that, okay, after 2 cycles of selection, I should get some gain of this selection. But sadly, I didn't get any gain from the selection from these 2 years of cycle selection. So the y-axis is the percent the sterility. The higher the bar, the more susceptible it is. So the lower, the more resistant. But across the cycle 0, 1, 2 here, there's no significant improvement on this. So I didn't get any significant gain from that or significant realized heritability, which means that the 50% of my PhD time that has already devoted to this has nothing. So I stepped back and I didn't cry. But I stepped back and try to look at the bigger pictures. Okay, I may think too narrow to just few cultivars that I tasted only couple cultivars. So last year I had a chance to access to the association panel, which consists of 509 switchgrass that has been genotyped already, and it was transplanted in Ithaca last year. So I have the chance to went to the field and do the field evaluation just roughly. And as it showed in the segregation, and we see some variation in the lowland type and upland type switchgrass. So based on these results, I really hope I get significant peak from genome-wide association analysis. But again, no significant peak from this. Even though the QQ plot shows shows no significance at the end here. So I would try my last year of my PhD now, focus in this summer to try to phenotype this trait again. By now, detach the leaf from the field and put it back in the green in the lab and spread it and evaluate the disease later. So I have 5 minutes left. Let's moving on to something impressive. I have another disease resistant. Do you remember the disease resistant to Bipolaris seed rot? Which basically I didn't expect much about this. So because I just soaked the seeds in just water and tween as a control, compared with the seeds with the treatment, which is an inoculum. And based on that, I collected the data based on germinate or not, 0 and 1. So based on that comparison, I can also calculate the percent germination reduction, which I can turns out that I can calculate heritability based on hapsip. However, the way that I make the selection, I make only a selection based on individual plants that it germinate from the treatment one. So I need to find a way to calculate the heritability based on individual selection, and I found a way to fit the model in binomial model. And luckily, I get pretty impressive, moderate to high heritability based on hapsip selection, ranged from 0.68 to 0.9 in each different cultivars. And let's focus on the individual selection here. That I get in Kevin Rock that I make the selection. I had the 0.3 heritability, and which is also significant heritability. So I really have my last hope in my PhD program that it may work, and this result is showing the progress of my selection. The x-axis is percent germination, and each pairs of the bar representing each cycle of selection. On your left-hand side, it's a Kevin rock, and your right-hand side is a shelter. So let's focus on the parental line, parental generation, the cycle 0. You see that the blue bar, which is a control one, has significantly higher germination than the red bar, which is a percent germination of the treatment one, which is consistent with the shelter here also. That is significantly different. However, within only one cycle of selection, I can reduce that gap between the blue bar and red bar to non-significantly different. And based on that, and based on this gap between the blue bar and red bar, I plot the next graph, which y-axis represents the percent germination reduction. However, I didn't see I didn't see any significant progress in caving rock from cycle 0, 1, and 2, but you see that the trend is reducing, which means it's more resistant to the disease. When we consider the shelter one, from the the parental line, the percent reduction is pretty high, but only one cycle of selection, it totally reduced that one to improve that a lot. So based on that, I calculate I get a significant gain from this trait up to like 100%, like in one cycle of selection, which I need to repeat this. I already repeated a couple times, but I need to repeat it again. But it's pretty impressive. Only one cycle of selection can do that. And I wanted to compare with my individual based heritability, but because of the trait that it's only 0 and 1, I cannot calculate the realized heritability based on that. So I found a way to calculate the liability heritability. And you see here at 0.4, it's pretty close to 0.3 that I calculated earlier, which means that, okay, I got pretty like consistent results from this selection. And now let's compare these two resistance. As I presented to you earlier, I didn't get any impressive gain from resistant to leaf spot. That's all flat here. But you see the trend in resistant to seed rots here. It's some improvement here, and so based on this data, I can calculate the correlation between the resistance, and as the graph suggests, there's no correlation. It's non-significant here, which can explain that the mechanism behind the resistance are different, which makes sense, right? The resistance in seeds and resistance in seedlings are different stage, so it has no correlation. So in summary, due to the low and non-significant for the resistance to bipyrate leaf spot, I didn't get any gain from selection. And this year, I'm gonna try the GWAS analysis again, like improving my phenotyping in the field again. And I hope I can come up with some mechanism behind that, some significant peak behind that. And based on moderate to high heritability of the resistance to bipyrate seed rots, I get pretty, although I didn't have like a significant gain in caving rock, but I get pretty impressive significant gain in shelter. And I'll continue the selection of the third cycle again. And based on the different trends in this resistance, we get like non-significant correlation between these two traits. So thank you for all of my lab, and thank you for your attention. Very welcome for any questions or suggestions. Yeah, so I calculate the broad sense heritability, and even the broad sense is only 0.3 for that. So I need, and because last year it was not a good representative year, because it's pretty dry, and it's not it's not good for my disease to occur in the field. And also the plant was so small, it was just transplanted, so it's like this size compared to like this side of switchgrass that it should be. So. What is the ploidity? Ploidity, so it ranges from tetraploid to octaploid, and the one that I make the selection is all octaploid, so it may like have some like difficulty in terms of like pushing those genetics to. What program have you used for GWAS? Oh, I try like couple of them, but the one that I presented to you, I used just only R by using RR block package.
Jeff Endelman has a GWAS which you can use for
Oh, the point in this case is I try RR blob and tassel, because switchgrass, although it's octaploid and tetraploid, but it has a disomic segregation, so we can like use those like pathway. Yep. Thank you so much.
This has been a production of Cornell University, on the web at cornell.edu.